Hello, this is Dr. Srijip Karmakar, a National Postdoctoral Fellow at the Department of Bioscience and Biotechnology, IIT Kharagpur, and this is a short video of my current research. As a postdoctoral fellow, I'm working on in vitro and in vivo investigation of biomolecular condensate formation by Hox and tail transcription factors and their role in leukemia. I will briefly explain what is phase separation and why are we interested in the Hox transcription factors. Phase separation refers to the transition of a homogeneous single phase to a two-phase system. Like oil and water, homogeneously mixed together will separate into a water phase and an oil phase over time. Similarly, proteins also phase separate from their homogeneous aqueous environment and form protein clusters known as condensates. Not all proteins phase separate. The intrinsically disordered proteins or IDPs are known to phase separate into condensates. Hox transcription factors are also largely disordered proteins with a small folded domain called the homeodomain with which it binds to DNA. As transcription factors, Hox regulates the body patterning during fetal development, hematopoiesis in adults, and have been found to be associated with diverse cancers where aberrant expression of Hox has been observed. Therefore, we are interested in the phase separation of Hox transcription factors and the implication of it. Through large-scale simulations with coarse-grained models of the disordered regions of Hox, we observed some interesting phenomena taking place. At first, the relaxed structure forms compact conformation from where the terminal disordered ends protrude out like a tadpole. These tails from different tadpoles interact transiently, establishing strong contacts over time and pull each other and fuse. In our large-scale system, several Hox chains fused likewise and formed a large spherical cluster showing condensate-like behavior like liquid-like mobility and boundary retention. Our key findings are: 1. Hox transcription factors have high propensity to phase separate. Second, phase separation takes place via fly casting mechanism, which is the interaction of the terminal ends of individual chains. We also found that short linear motifs in the disordered regions form transient folded structures, possibly indicating towards recognition sites for sequestering other proteins in the cluster. And these motif maps have been curated in an interactive web page called Hoxverse, available at the provided link. So this work has been published in 2025 in the Journal of Proteins, Structure, Function and Bioinformatics, and presently the experimental verification of the Hox phase separation is being conducted. Apart from my main project of my NPDF, I am also involved in two other projects. One is protein prosthetic group reorganization into supramolecular assemblies of amyloid quantum dot systems. In this work, we are investigating the formation of quantum dots from proteins containing prosthetic groups. We have successfully developed heme quantum dots from cytochrome C aggregation, where heme dissociates from its binding pocket due to induced misfolding of cytochrome C, followed by geometric stacking of heme molecules, forming extremely small nanoparticles with fluorescent property. Now, these quantum dots have been observed to enter mammalian cells without any transfection agent or carriers, and they localize in the cytoplasmic zone. As a result, in confocal imaging, the fluorescence of these quantum dots could produce clear images of the cells, indicating potential application in medical imaging and cell organelle targeting. This work is currently under review in Nature Portfolio's Communications Materials. I am also working on the development of a point-of-care sensing platform where specially designed peptides having the ability to recognize specific proteins or RNA or DNA are attached covalently on carbon nanotube surface, where binding of the analyte molecule results in a change of current response, which is read as the concentration of the analyte molecule. This is intended to develop a point-of-care device for liver function test.